So this morning, I went to breakfast and everything was okay, and then there were some issues to resolve, and then suddenly I realized, because I was thinking about the project one of the groups is doing on the dictyostelium, and I thought, it might be really good if they put this into moving cells. But for that to happen, it would be good for them today to hear about the biophysics of cells. So I'm going to change a little bit the order, the sequence of things. So for something totally different now. Yesterday we saw a little bit about the auxin modeling in roots, right? What I was going to continue telling you, and I will do that later, is how we can follow up with like hypothetical experiments. What if things had, what if different mechanisms had been selected for, what would be the consequences for development? So it's still something I really want to talk about. And this afternoon I'm going to give a seminar here for the Institute, but also for you guys, because I'm going to touch back on lots of things we saw and start introducing more about how we start looking at cell polarity and tissue polarity. But so today, we're going to actually start thinking about, if we really want to take patterns in biology seriously, at least at the level of organisms, organs, unicellular organisms, which is quite a big part of biology, well, the building block of life, we might want to start thinking about the biophysics of these cells and tissues, yeah? So I'm going to talk about something called cell surface mechanics. So for cell surface mechanics, and this is a very broad concept, we just sort of think about, first of all, just think about a embryonic animal cell or amoeboid. So it's going to have, right, a membrane. Yeah, you guys have been talking about these things quite a lot in last week. Inside, right, it's going to have some type of jelly type of substance, the cytosol. And there'll be some other things floating around, right? A lot, yeah? We also know that there's going to be cytoskeleton, right? And the cytoskeleton can apply forces and projections from inside the cell, so that cell, that bag sort of moves. Have you guys ever been in those balls in the water? They put people in like these giant balls. Yeah, so you can move from inside. You can't poke yourself out. It's a pity, but there we go. Okay, so that's the cell. So what we want to describe is that a single cell will have surface tension, yeah, and that can be altered, like biologically, by adhesion, yeah. So how sticky this will be with whatever it's in contact with, okay? So adhesion. We're going to talk about that. So that free energy here. It also has possibly some of them, not all. It might have underlying here this membrane. There might be some type of mesh structure. Okay, so this is like sort of like a network, and that mesh structure doesn't want to give or go too easily. So there might be some type of perimeter that it likes to have. Like a rest perimeter, okay? So we have like a target perimeter. So we talk about things like target perimeter. Okay, this is a very optional term, I can tell you. But I'll show you where it gets handy in some cases. And obviously the cell, it's not just like a bubble, right? As you guys learned last time. Because it can't just, if it's under tension, it's not just going to, let its area go smaller because its gas is leaking out because of pressure difference. It does have the possibility of things to flow in and out, but this is regulated osmotically, osmotic pressure, yeah? But it has a rest point, it has a target area, okay? So that's it. Those are the components that we need for cell surface mechanics. So all the models, they can come in different flavors. You can have vortex models, finite element models, right? They can be continuous, but they're going to have those ingredients. Adhesion, right? Surface tension, perimeter, some type of perimeter rest area, which can be dynamically updated area, and so forth. Okay. So, always a nice context. So this is the hydra. This is how I got interested in biology when I was a student in physics in URCS. And we actually, this is a Brazilian hydra, right? And what we used to do, so this is super, super fun experiment for physicists, like you can chop it up in little pieces, you take lots of hydra, chop many of them up, then we actually, that was like, oh, really, really, it seemed by that in those days really like, wow, trypsinate the cells. That means you make sure that they don't adhere to one another, right? So they're isolated in suspension. We put them in a little centrifuge and they all collide and you get this little lump, yeah? And this lump is like lots of different cells together. And the funny thing about this hydra, it only has 2 tissue types. It's like a sock, like you know, when you put on a sock, but a double-layered sock. So you have an inner part, which is the endoderm, yeah? Because these little critters here, they have like a sort of a sticky foot, and they have like an opening up here, which is their mouth and anus. Okay, and then here they have the ectoderm. Yeah? And in the middle they have like an acellular goo, the mesoglia, okay? But basically, cells can be ecto or they can be endo, like in that structure. So we have a mix of both of those cell types here, but then lo and behold, after a few days, the whole organization has reformed. And I will show you some pictures, but from total chaos, because we just put it all together randomly. Whoopa. Oh my gosh. Okay, that was that. It starts becoming circular, and you start getting like things like this. They start percolating, but these guys move out, and the yellow clusters of cells, they start clustering, percolating, and they move in. And then with time, they allow some water to get in here. So you have like a football. So so you have from a porridge to a football to a sock, okay? So we were interested in this here because from here to here, some other stuff started happening. And that's interesting because we know at that moment that there's not much cell division happening. So it's cell sorting. So cells are actually moving. Hey, you know, let's stick together, and they do that stuff, right? Now, I'm sort of telling you, yeah, so here, those were the movies we were making. We were also interested in this little thing that it does, like it sort of pukes out stuff. It's called bursts. And that was also interesting because, yeah. Okay, so that was it. So we have, now let's go back to what we know, what we heard last week from Evan. So we have the rounding property, like which is surface tension driven. We can have, in this system, we can have something called engulfment, right? So basically one tissue will surround the other one, right? And this is happening through this phase separation. So like, you know, when you, I don't know, mix water and oil, you get all these droplets and suddenly they separate out. So it was very reminiscent to this idea that this might just be cell surface, like surface tension driven, yeah? But where is the surface tension? How is this working? So that's what I want to talk to you guys about. Well, that surface tension is of paramount importance for biology was said by many people, and Darcy Thompson included. He was a Scottish naturalist, and he has these thick books, and in it are many pictures of tissues, of cell configurations, walrus tusks, how snails look like, the horns of goats, everything. And he was looking at everything and saying, hey, this is a great example of surface tension. I can actually mimic this if I take this gooey thing and I do that, you know. He was just showing how you could derive these things by a combination of growth and having different properties for surface tension. So it's really great. So a lot of modeling is done by that, but usually here we consider things more for these scales like as a continuum, right? So we really want to take a look also at that foam part of biology, the fact that we have these cellular structures and not just a continuum phase. So yeah, so this is sort of a little bit better than experiments that we were doing. So here you have 4 hours of the hydra. We're looking at these different cell populations, and you see how they percolate, and they start then forming out these endoderm and ectoderm separated layers. It's very nice, these experiments, because we get time scales. We can see how quickly those percolations happen, and that tells us something about if we are assuming that this is surface tension driven, what's happening behind the scenes with the effective energies. So we want to take as the building unit our cell, and in this talk, I will consider cell perfectly homogeneous. There's going to be no front, back, no difference, no transporters, right? It's just on all sides the same. Okay, so to understand this phenomena of cell sorting, it was proposed actually by a biomedical researcher, Steinberg, that cell sorting that they were seeing could be explained as driven by differential adhesion between cells. So cells, when they enter in contact with one another, they have these like lock key things here. So they have molecules for adhesion. They're called cadherins. Yeah, those are the biggest class. E cadherins, I cadherins, like cadherins. When you have them sticking on a substrate, usually the substrate has to have something, some fibronectin, some type of, they will then stick here with other type of adhesion molecules called integrins, okay? So they can actually regulate very well how they want to adhere with cells and how they want to adhere with surfaces or linings in your body. And the cadherins will come in different flavors. So they can have like cadherins to stick more with yellow cells, and then a different type to stick with the blue cells, for example. Yeah? And remember, adhesion is something that two cells do together. So it's impossible for a yellow cell to adhere well to the blue and the blue not to adhere well to the yellow, right? So you can already see that when we have these adhesion matrices, we're going to have these symmetric matrices coming out. Okay. So cell surface mechanics, as I told you, have these. So basically what do? We write down an energy function. Yeah. We used to call this a Hamiltonian in the past, but after so many discussions, heated debates, we just call it energy. And so the perimeter of the cell is something which will be in these models, in this, will be altering, right? And so will the area. But these are the parameters which you give, which are given, the fixed parameters. J here represents the adhesive strength. People just sometimes call it adhesion. But remember, it's a cost. It's going to be a cost here. The higher it is, the higher energy, right? The idea is that cells will then minimize. We're going to minimize this energy, right? And then see what type of configurations we get. And so the adhesion is happening all around, proportional to the perimeter. So it's a length. So this is P, so J times P. Yeah, that's that first term, right? Are you guys okay? Okay. So now these other things are like your Lagrange multipliers, your constraints, right? Because until here, we're basically just describing foam. Now we say, okay, it could be that you have that stuff here, this cytoskeleton, cortical mesh, right? So cortical tension, so you might have this target perimeter, so that's this big P, right? And so we have a constraint around that, squared. And we also have, and so we have a lambda P, this is the Lagrange multiplier, and here we have lambda A, and then here we have our target area A, which is the desired area that that cell would want to be at, and it's going to fluctuate around, so the little A minus A squared, yeah? So these are the two things. I would say about 80% of the time we forget about this term, and just this one. But this is it, this is all there is. Yeah? Okay. That's it, actually. Okay, but now it gets a little bit more complicated because there are things happening. So we will take a way of simulating this, of looking at this, by simulating it on a grid. So we take our cell, and this is like, there's a group here which is working with an agent-based model of excitable media, where every little pixel is a cell, basically, doing something and moving around. The problem with people in the past, you know, there was a lot of theoretical biologists that have been working with trying to get cell sorting happening. I don't have these slides, but it's really comical. One day you should read those papers. They were trying to get in a CA universe, where you just have the grid, right? They would say, oh, you know, we have a population of blue cells, right? And yellow cells, and now we're going to make some simple rules like, oh, how they hop around, and they're going to hop less frequently if they're next to a neighbor that they like, yeah? But what happens when neighbors start coming together? How do they hop? How do you hop a cluster? Right? You have to have another set of rules for a cluster of 2 or 3 or 4. It becomes very, very difficult to do cell sorting. In fact, their models became bigger and bigger and bigger. Draconian is the word. Yeah? And what was their conclusion? That Steinberg was wrong because it was very difficult to get cell sorting, just due to adhesion. There would need to be longer distance things being felt. And that's because this is just, I mean, they were really smart people, but you have to be ever so careful not to get toggled out in the formalism you're looking at because, you know, those artifacts are just there because you're looking at that. But the moment that you start thinking that a cell is not just a pixel here, not just one grid, but could have many, right? And you can allow it to change its shape and feel things at this boundary, you have 2 scales. You have a scale at the perimeter, at the edge, local, but you also have a bigger scale. So cell shape changes would be very important. And we're going to take a look at that. So this is not how it really looks like on the computer. This is like a really bad drawing that I made once, and I just, you know, like, as in development, sometimes you get stuck with things, and you just have to live with bad pictures. Okay, so here we are. So this would be the grid. We give it numbers. These numbers are the spins, right, or the indexes for your cell states. Yeah, so the sigmas here. And so this is actually, this is very much thinking about like Potts model, yeah? Now think about Potts model. And you're going to look at your neighborhood, right, for your interactions. But you also will have these 2 terms in the energy function, okay, which will tell you what's happening with that sigma if your area gets a bit bigger or smaller. What happens to, you know, to the energy of that cell if it gets bigger and smaller. And then you sum that up. You go over the whole lattice. You sum it up for all the cells as well, and you have this total energy. Yeah. Okay. So every number index is an individual cell. But now we want to put biology in, like endodermal cells, ectodermal cells, because we know they're different. They have different coherence. Yeah? So, okay, so these are the spins, these are the states, these are the individual cells. And we also have like zeros that we like to keep for medium. We could have other type of things which don't have then area term, which aren't like, could be just mesoclay, other stuff, right? Okay. And I didn't put the other picture in. Shit. And then you can start saying, okay, cell 3, 4, and 7, you know what? They're the green cells. And cell 46, 6, 2, 9 are assigned to the yellow cells, whatever. You can assign them then, right? Every cell will be assigned to a type, cell type. Okay? And now I'll tell you something. There's something very confusing in this whole field. I talk about surface tension here. I'm going to start calling this interfacial tension. I'll show you why afterwards, because we're going to think about what happens when we make a little blip or a little thing of the cell. Okay? So sometimes we can talk about that energy of the cell, but at another moment, sometimes people might be talking about a tissue composed of many, many cells, and what is its surface tension with the medium, yeah? So this would be like, I can think about what's the surface tension here of my cell A, or interfacial tension of my cell A with the medium, yeah? If I don't have a perimeter constraint, it's just going to be J, just my cost. But here, what is my surface tension of my whole aggregate with the medium? This will be a combination, as you heard Erwin talking, about these bonds minus the connection, the heterotypic bonds. Yeah? I'll show it to you later. So when people start saying, oh, surface tension is this, surface tension is that, it might just be because they're either thinking about a collection of cells or they're thinking of the cell. Okay? Please interrupt. I know what I'm talking sounds obnoxiously simple, but it really gets people very confused. People even in the field. Okay, so then we do a Monte Carlo simulation. So we have defined energy for a configuration that we think is agreeing with surface mechanics. And now we want to have the dynamics of that. We want it to start moving actually to relax to a minimum configuration. That was the original idea. Now we don't think like that anymore, but there we go. It's a little bit heretic for me to say that. But okay, so you have initial configuration. So I'm just zooming into some spins here. And we know that configuration energy E1. So we're going to randomly choose a site. Then we're going to randomly choose a site in its neighborhood, like 5. And then we're going to ask the question, what if 3 extends into 5? Okay? Then we have a configurational change, and we're going to have energy E2. And the nice thing for the computer, it doesn't have to calculate the whole thing again, right? It just calculates very quickly that little change, yeah? Because that's what we want. We want the difference of energy. So CPM simulations are so fast if you do it right. And now we're going to ask, is this difference E2 minus E1, which I will call here delta H? Okay, for a moment, imagine that this Y is a 0. Okay, just easier. That would be your traditional multicolor. So if your energy change is negative, so you've decreased energy, you accept it, yeah? Good, it was good. P change 1. If it was, however, positive, we accept it with this Boltzmann probability, right? So we have here, the bigger the energy, the less likely, and the bigger the temperature, the more that you accept those mistakes. That's your noise, yeah? So happy to be in a public that I don't have to respond to the question, if I do my experiment at a different temperature, like, no, it's not that temperature. It's, okay, I'll tell you what temperature this is. So the temperature, this temperature here is the acceptance of a cell to go over a little energy, right? So that is the activity of the cell. So cells have ruffling, right? And this ruffling is active energy, in fact, right? And so we can actually even change experiments, but it's not the temperature. We change how much of those undulations a cell is actively doing, okay? Now, the Y is introduced here because in things like dictyostelium, when we're going to model this after tomorrow, when it forms a stalk, it actually has to be a little bit more rigid, or it goes down. So we put this Y here as a yield. We say that it doesn't only have to be favorable, it has to be more than so much favorable. Okay, so it's just a bias that we can give for different cell types, and that will really change the stiffness of materials. Which is really interesting, because this is not entering in the energy function. It's entering in the update, so. Okay, so this is the foam physics of cell mechanics. Yeah, so this is your simulation. Every cell is of the same type, of course, you know. Underneath, they have these little different spins, but we don't care about that. We just care about their type. That wasn't me, was it? No, please, no. Okay. Okay, we have to restart. Very temperamental. So I'll have to dance it, right? So what you saw is they're like having those little ruffling things, and they start. Now, when two aggregates come together, the neighborhood here, when you look at that energy function, you sum up the J's over a little neighborhood, right? So if you have a cell, like this is a cell, yeah? So I'll take this here, and I look actually at the J's in a little neighborhood. Sometimes first neighbor, second neighbor, the little neighborhood, yeah? Because there's also the temperature. So it's not that it feels that there's another cell somewhere. There's no chemotaxis, there's no anything. So it's just because you're moving around a little bit. Once you do make a contact, you know, that contact is so much favorable because you have adhesion, right? So it's that you zip, you quickly zip. And if you doubt that this is happening, if you ever try to wrap a present, because I have to wrap presents now for Christmas for my kids, it's really terrible because we have these like, fita adhesiva, the tape. When you put the tape up, it tries to tape it. The moment that the tape falls on itself, it zips itself up, right? So this is sort of the mechanism that happens here. So once it touches, you see it zipping. Okay. However, then some people might say, yeah?
Touch each other, how you can define it, because we use this information to calculate the adhesion between different cells.
Yeah.
So how we can measure it?
Okay, okay. I'm so happy because I like to tell how you measure it, but nobody ever does. They don't even get why I'm talking about that. It's the first time that someone asked the question. Okay, so when I talk here that, for example, you know, I mean, look, the energy, the first term here was J times P. Yeah? Simple enough, right? Area is very easy. But what is P? What's the perimeter of something on a grid? Because, you know, I had this discussion once with a mathematician. He was like this. He was telling me that in the cellular POTS model, everything should just become, if you have first neighbor interactions, a square. Because it has a least perimeter, right? Okay, because if if I have 4 like this, or I have 4 like that, you know, I'm going to have a very different type of perimeter, right? So how do you calculate perimeter? And I will describe it. I have the calculations done, but then I also have like a lookup table that when you change perimeter in your simulations, you have to renormalize it to the true perimeter, depending on your interaction energy for calculating your adhesion. So do you know when you calculate your adhesion? You look at in your neighborhood and you say, oh, how many J's of this type, of that type? You sum it up. That neighborhood is something that you'll need to, if you update the neighborhood, because sometimes you say, oh, I just want to make the interaction a little bit bigger, a little bit smaller. But what you're actually doing is you're changing the perimeter of your cell, the true perimeter. So then, if you want to then go back to, no? Oh, no, really? Oh, thank you, that's so bad. Yeah. It's my fault. Okay. Could you put this in? No, it's my fault. I hadn't. You see, usually gives me a warning, but it was I couldn't see it, of course, right? So, yeah. Okay. Oh, it's this one. Yeah, I also forget that. Yeah, could you? Thank you so much. Yeah, so let me tell you also that if you don't do your perimeter correctly in your calculation, not only will you have a mismatch with your cell surface mechanics, any predictions that you have, you won't be able to compare the cellular POTS model with any other model. So what we can do now, like you can look at a vertex-based model, take those parameters, plug them in. You do some adjustments given your grid properties, and you have exactly the same configuration, dynamics, and so forth. It also means that there's some, you know, did anybody ever do a cellular POTS model simulation, or, yeah? Do you know, sometimes you think, oh, I don't want to wait too long, so I'll make my grid size a little bit chunkier. Or now I want to do the same simulation, but really nice, small grid. But how do I do that without changing the dynamics, without changing the, right? So then you also have to think a little bit of what that means. Okay, so my computer is back alive, just a second. And the reason I think it's good to spend a little bit of time, it helps us understand cell surface mechanics, but understanding well the cellular POTS model is really good for, because I think it's a very powerful method for morphogenesis. Just a second. You took, yeah, whoa, don't touch anything, okay. So here I see it, so that sounds good. Should I go one back, maybe? Just like... Yes, okay. Okay, so to the left is the CPM. To the right is something which I like to say, like, it's when you put your statistical glasses on. And so what you see there is where the spin flips are happening, where the changes are happening over time in a moving window. Yeah, so you see the density function of where we have your changes. Does that make sense? So what you should appreciate there is that you don't see the grid anymore. So if you take those simulations and you look at the angles and you look at the distributions, you don't see any bias to the grid. You'll have perfectly 120 angles predicted, right? So all of the Neumann rules will be observed here, yeah, as it is. So it's a very nice behaved system. Okay, And in fact, that is sort of where the perimeter is, right? Not there, but there. So we're going to see that soon, mathematically. Okay, so let's see if we can now, I mean, you know that we can do cell sorting with CPM, but this was the history, right? Can we do cell sorting just using adhesion without anything else? And actually, Glazier and Grieneisen, François Grieneisen and James Glazier, they did. They showed that it's possible with the cellular pots model. They met in Japan, and they worked together on this, and they were able to show it for 2 cells. But what I'm just showing you here is that after that, some experimentalists, this group in 1996, they actually were able to measure the cell surface tension of tissue aggregates. So to the left, you have the aggregates that are isolated from a chick embryo. So you have limb buds, epithelium, I don't know what epithelium exactly, heart, liver, and neural retina. And so you take the aggregate, and maybe you guys know these experiments. You put the little aggregate, you know, which is, it's like a, you know, it's going to be a rounded ball, like a meatball, yeah? With lots of little cells. Yeah, what would this be? Like, I don't know, 600 microns or something like this. Like, they can be small, they can be bigger also. You could take one like of 1 centimeter, like you could even take a big one, microscopic. Anyway, you put it between 2 plates. You make a piezoelectric. You put the force, and you let it, you squish it, yeah? You squish it. Now, you don't let it, you immediately start measuring the force of that squeeze, and then you can measure the surface tension. Same thing that you would do with the oil droplet, okay? Obviously, if you leave it long enough like this, we will have rearrangement of the cells, and then you're going to have a plastic response, and you're going to have different physics entering. But you just want to have that first little squeeze, get the surface tension out, so you actually have something here, right, which is dynes per centimeter. Okay, so the limb bud, as you see, has a higher surface tension, that tissue type, than the pigment, which is 12.6, epithelium, 8.5 for heart, 4.6 for liver, and 1.6 for neural retina. Now, what's really cool here, they stained them also so you could see them, is that when they do those experiments of aggregating those cells together, they will sort out. And do you observe, it's always the one with the highest surface tension that ends up in the middle, and the one with the lower surface tension that will engulf. Yeah? So at least the predictions of this hierarchy, right? And what you see also is, for example, and this was also really important, it's not like that the heart tissue wants to be in the inside. Because if you put it together with the liver, it goes to the outside, right? So it's really just to say there's nothing conscious here. The cells are not moving in a certain direction because they have neighbors. They just, passively are doing their wiggly stuff and end up somewhere. Okay. So here is that same situation. We give the surface tensions as measured into our simulation because we can calculate back the combination of adhesion energies that have to give precisely those values. And we can mix all the cells up together because, hey, it's a lot more easy to do simulations and experiments. And what you see is over time, we will have the hierarchical cell sorting out into the tissues with the lowest surface tension tissue in the middle, the other ones around, and so forth. Yeah? Now, what you can already observe is that it becomes more and more difficult, like that green blob has to find that other green blob. It doesn't know of its existence, but it's always having this temperature, these fluctuations, right? So you're always allowing it to go over those energy minimas. And at a certain moment, even when a little part will touch, yeah, look, it'll zip. Doop. Yeah, and that's why it's so good to have cell shape. And you couldn't do this with distinct points, right? Okay. Even the yellow guys at a certain moment wanders in. So, cell adhesion is sufficient to get cell sorting, right? Now, some people were really like, no, it's not only you need the flux. Okay, so this is a simulation with 0 temperature. So yeah, you grow because you're just going down your energy gradient and then you stall. So if you have no temperature, you're just going to get stuck. Yeah, so really important is that you do need to have energy for these things to happen, like you need fluctuations, temperature for these things to happen. So in a system like this, temperature is extremely important, yeah? And it's helpful, right? Patterning. I mean, you guys are physicists. But what was really nice is we were working on a project, ERC project with engineers with robots, little robots. And they wanted these robots, like if they have division of labor, to suddenly cluster. But they wanted it to be in a way where you wouldn't have a supercomputer, you wouldn't have a pre-pattern, right? And I said, hey, why don't we take these biological examples? So one thing we did, it's not published yet, it's called the cellular bots model. So we have, because they radiate a local light source, and they can actually know how close they are from one another. Now the problem with these robots is because we wanted to have something that would work with a swarm of robots, not just a few. So they're quite cheap, they're quite imprecise, and they make a lot of mistakes. So even doing something perfect is the noise, because you have the inherent noise of their imperfection. And the more, the merrier that it works. And actually, I mean, they shouldn't be completely kaput, but, being not that, having those interferences is actually helpful. And whereas the engineers were coming with solutions where they're saying, oh, but these robots aren't good enough, these robots aren't good enough. And so then I also learned that it's very different to do robotics on a computer or actually do them with the live robots, because you will never capture all the variation that you have in the hardware. Yeah? Anyway, so I think that biology, morphogenesis in biology, even something as simple as cell sorting, has a lot of things that we can learn from. Okay, so now I told you that, like, let's say the green population of cell there has an adhesion JA, the yellow has an adhesion JB. So this is the type, this is the adhesion energy that I give for that type. And I also will have to say, what's the J between AB? Oh, I'm sorry, I should have been better here. When I say the energy of A, I mean, what's the surface of A with a medium? Yeah, you always have a medium. What's B with the medium, and what's the interface between A and B, yeah? So that's this J here. Now, we can then calculate exactly as, was explained by Evan, we can calculate the gammas, so the surface tension between the tissues. And I don't have it. So, JAM, I'll get a better color. Is JAA. It's adhesion that it has with itself. I'm sorry, we also have yellow with yellow and green with green. Yeah? Yeah. Minus JAM over 2. Okay? So this, oh, yeah, J. Good. And so let's do a JAB. This is correct. It's the heterotypic minus the homotypic. Yeah, sorry. Yeah. Definitely. Yeah? Correct? So this one here will be the JAB. So how much is that energy? Minus JAA plus JAB BB over 2. Okay. So heterotypic minus homotypic. That gives you that extra surface tension. Oh, it is up there. Okay. So good. So also, the angles that those tissues will make with one another will be given then by the constant. So you just have to, I'm not even sure if they sum up, right? But they will be at equilibrium. And there you can already see that if one of them is bigger than the sum of the other two, yeah, you're going to get engulfment. One of those, the triple point will go to 0, and one of those phases will disappear. So you can get all the properties out just by looking at those three arrows and their angles and how it would look at. Now, let me tell you, you can do this with, 2 tissues with 3 tissues, doesn't matter. But what's also very funny is that if you really zoom into what's happening at the vertex, the cells might have a different angle themselves, because there the J comes into contact. This is a statistical property over all of the cells themselves. Okay, so basically, rule of thumb, if we have a negative surface tension of your type, you're going to get your cells separated, so you can have simulations with single cells. What would happen if you would have a negative J? Just a negative J. Yeah, your cells would disintegrate. Okay. Unless you put a perimeter constraint. Okay? So there we go. And then we can start looking here, right? So if you have positive surface tension, you'll want to aggregate as a tissue. And then depending on how your tissue surface tension is in relation to another tissues, you can get checkerboard patterning, which is very common in brain and in the retina of humans and chicks and stuff like that. And you can get partial engulfment and all different things happening. Yeah? Okay. So this is not about cellular parts model. This is just about thinking about patterns of these cells. Okay, so I'm going to go a little bit faster now. So how do we link these results to other models that exist? So how do we now translate these parameters and this very simple Monte Carlo way of doing things to actual cell surface mechanics? And that goes back to your question, what is the perimeter? And the other question I just wanted to address is, these simulations, they're actually really nice and beautiful, and we can get lots of interesting insights, but how can we actually link them back to experimental measurements? Yeah? And for example, what do we do with mechanics of tissues? So one of the problems now of linking it with other formalisms is that we live on the grid and other formalisms generally don't. So one problem that people have a lot, or that if you work with grid-based models, is that you don't want your things looking like Minecraft, right? They're sort of cute, but this is not the type of biology we want. So just be mindful that, yes, indeed, here we have like 0 temperature, so we anneal it, and this is the temperature below the critical ruffling temperature, and this is above, right? So there is something like critical ruffling, which will have to do with what your J's are and your neighborhood size, and you just have to make sure that you're above it, right? That you're not, because your cell surface tension is anisotrope to the grid as long as the temperature is low. It becomes totally rounded if the temperature is high enough, right? And you guys know this from crystals, those wolf constructs where you have these beautiful flowers and we could predict what crystal you would get and how you would round it up, so the same thing. Now, it's very dependent. There's a compensation, how much temperature do you need and what's your first nearest neighbor on a square lattice. You can do simulations on hexagonal lattices. We love doing that, actually, for cytoskeletal reasons. I'll talk later. Again, are you above the roughening temperature or not? And again, remember, sometimes your cells look a little bit weird. If you take a snapshot, they look like they're boiling. But their mean cell shape is actually very circular. And then you can go to higher neighborhoods, and you can just see how these things work out, yeah? So first rule, just be mindful that you are at that right point. And this is, again, for 3D. We can show these things. We have graphs that show actually what the ideal number of neighbors are and what you should choose for, So we have a lookup tables for that, just to be in the right regime. Okay, now the other thing we want to start thinking about is, okay, we want to actually do some physics now, right, with these equations. So if this is this energy function that we have, right, with these terms, what is the interfacial tension? So I want to ask the question, if I would what's the partial derivative of your energy in relation to your perimeter? So if I make a little blip here, a blip here, what do I get? So you take this here, you take the derivative, partial derivative to perimeter. I only have 2 terms with perimeter here, the J and this one here. So then I have the 2 lambda p, p minus p, yeah? So what you see here is you're going to have a constant, which is like these parameters coming together, and you will also have here a term which is dependent on your perimeter. This is the interfacial tension. Now remember, if you are not having this term in your model, which usually you will not have, it collapses. Your interfacial tension is just your J. Okay? However, if you do have a negative J, this is bad, your interfacial tension is bad, and you're going to diffuse around, you're going to, right, as you guys predicted. But if you do have a negative J, you could compensate that by changing these values. There's nothing magical about a negative J. You can, you can always get rid of that, because it's about the interfacial tension. The pressure, right, it's minus the partial derivative of energy with A, so how much pressure inwards do I get if I do a little inflation or if a little compression, right? So then you take the derivative with area. The only term I have is this one, so I have this term here. And then you can think about, okay, if I look at the gradient along the membrane, if there's a little, what would be the force if I take the gradient of this energy? And we will get one term which will be here, gradient of the area and of the perimeter. And I'll come back to that as well. So the nice thing here is like people say, like, oh, force models are the way to go. Because you're doing something wrong because cells don't send energy. I don't know, they say things like this. But what we're saying is we're describing an energy. Every little thing that's happening there will have a gradient, right? Will have a force on it. It's just that we're not explicitly determining what those are, right? So now it begs the question, though, what is the perimeter? Okay, so this gray thing here is like a big cell. Yeah, so imagine a big cell, and we have these little pixels here. And when I calculate the energy of interaction adhesion, it's always done with a certain radius. I'm exaggerating a little bit here, okay? So let's say I have, I don't know, a radius of 5. So this pixel here is interacting with all these pixels here energetically. But the pixel inside also has this. And so does this guy, right? So you actually have a moving circle, and you will start counting. So you have to integrate all the overlap you have. So the way we did the math is like we approximated this here as a straight line. So let's say it's you have a really gigantic cell, a straight line. And this is the interaction at which you are summing up your J's of that term, right? And we are just considering, okay, we have these, right? And so you take those double integrals, and then you can calculate here this factor, which is what you have to divide the counted perimeter with to get back your true perimeter. And so it's really funny for 2D, it's going to be a radius to the third, and for 3D, it's that radius to the fourth. Okay? I can show you the calculations afterwards, but they're, yeah, okay, and then we're in business. Okay, what about the spatial resolution, right? What if I make, I have a cell, and I know my biological cell is, this is how we do simulations, yeah? We have a cell. I know my cell, it's a small cell, embryonic cell, 5 microns, okay? 5 microns. So maybe I have, 5, 1, 2, 3, 4, 5 pixel squares here. So I know every pixel here is 1 micron big. And why is this important? Because usually our simulations have something diffusing on them. So I also have a space scale, so I know a diffusion coefficient or something. So, but then suddenly I realize, it's getting a little bit too Minecraft. So I want to have a better resolution, right? So then I start chopping up these things, 20, right? So what do I do, though, with my J, with my lambdas? What do I do with all those parameters? Okay? You have to do this. So if you're scaling it, like 5 times bigger, so it's divided by 5, 25, 5 to the 4th, yeah? And there we go. You just have to do these parameterizations. I leave them up here for you guys. So this is to show you that we're doing it. So this is K1, 2, 5, and 10. So this is the resolution. So this is like very coarse, and this is very, very fine. And what you see is that we get similar like number of percolations happening, and we get the same sort of dynamics. Now, there's one thing that we can't normalize for, and that's time. Okay, and that's because when you are a little cell and I have a big block fluctuation, that's a huge fluctuation. Yeah. If I'm a really big cell and I have a small fluctuation, that's a really small fluctuation. So it will take longer for things to happen. but you still get approximately the same dynamics. But you would still have to think about a way of renormalizing your actual time scale. So there's a lot of discussions we have of what we could do better here. Okay. The other thing is, once you take this equation here, you can ask the question, I want to find a positive interfacial tension for my cell, because if a cell has a negative interfacial tension, it's not a cell. It's just, you know, it's just I put it in a liquefier. So positive, but I want to find the equilibrium for that. Yeah? So you can do a dynamical systems approach, and what you can get out of that are these, so this is analytic results. So we find actually two parameters, aggregate parameters, that can describe every single behavior of a single cell, given any type of parameter in the parameter space. So in region 1, your cell will have, actually, in this case here, it's going to be wobbly because it will have, it's still not exploding, but it's having a very unconstrained surface tension. Here, it's going to be rounded, and here, two things are going to happen. One is that, so this is this bistability we have. Here, you will always shrink to 0, so you can't maintain, your tension is too much, you can't maintain it with your volume pressure. You go to 0. And in this region here, there's a critical size that if you press your cell, it's going to deflate. But if it's above that size, it'll remain okay. Okay, so we have that transition here. And we can do exactly that same analysis in 3D, right? So again, we'll have just two parameters. And because this was analytic, we then looked with our models, like, okay, can I get, like, what's the single cell dynamics in region 1? So we have these fluctuating cells. In region 2, actually this is for the 3D case, we have circular cells. And then here we can get them disappearing. In one case, it disappears because it's just below the critical volume. Here, it's above the critical volume, so it grows. And here, it'll always deflate. So we can get that. Okay. So why was that important for us? And we did the same analysis. Let's look now at a cell in a tissue. Okay. And so how do we do that? How do you do this mathematical analysis of a cell of a tissue? Well, you suppose that it's going to be a hexagon, because that's how, you know, a cell would typically be in a tissue. So we calculated it for 6 sides. And we can find this region where it's going to be quite wonky, where it's going to be a perfect hexagon. And here, it's going to collapse. And here, you'll have the bistability. Like, you'll have that critical area. And the same thing could be done here for 3D. So 3D, we were imagining these shapes. Yeah, these are aggregate parameters of these parameters here. I can show you. I wouldn't be. We were able to.
I think these parameters can depend on the constraints of the model,
yeah,
like the target area or perimeter area, target perimeter too. No, which is depend on because the shape of the cell of this model apparently depend on the target area and target perimeter. Right?
Yes.
Or
so
the ratio between them, like something
yes,
like that.
Yes. So like here, do you see? So here we have the pressure is this. But in this case, for example, also, we have this target perimeter. It'll be in here. So yes, that's all together. Yeah.
So that phase diagram, when you analyze the collapses or these regions, I think which region would be depend on these two parameters, because if you
so those things, I think, to be honest, I think the target area is wiped out. The target perimeter is still in here. I can show you afterwards. But indeed, these aggregate parameters, they cluster them together. But the funny thing is, once you know like once you give me the parameters, I will know where you are here.
Okay.
Like that.
Okay.
Yeah? Mm-hmm. But yes, they will have an effect. Yeah, you're right. But why is this so cool? Let me show you. Do you see these dots here? This was a paper by Fahadifar et al., where he did, they published on a, what's it called, a vertex model, right? And you have to realize that the communities were like, okay, to do proper physics, you do this or you do that. Like people really think that they're doing different things because of the formalisms, although we have the same cell surface mechanics equations. But the funny thing was, we got these points out of their paper and we just plot them on top because they had also their parameters. So we took their parameters, we saw, how would their parameters translate here. And these were the lines, the empirical lines, the simulation lines they found that would divide the region between a floppy tissue and a stiff tissue and between disappearing and not disappearing. So 2 things here. First of all, we can show that, well, our simulations agree with this, but the analysis we do agrees with their model as well, meaning that they did really good job with their model. And that now we can also have a really nice way of talking between formalisms because we can see, if people are doing it correctly, their parameters we can immediately take, we know where we are, and we can put them in the CPM, okay? So that's super lovely. I mean, I find that like a trading zone. So I just want to tell you here about that positive J, because as I told you, the interfacial tension depends on the J plus your perimeter constraint. These are simulations where we only use positive J's. This is one where we only use negative J's, but we update the lambda p accordingly, so we have that same interfacial tension. And look, it's exact, I mean, we do use the same random seed to begin with, but think about it. The parameters are all different, but the aggregate parameters are not. So look what we get. I mean, all different, the J and the P. And we get exactly the same things coming out. So there is nothing special to a negative J as long as you compensate that with a cortex. Yeah, okay. Just because there are papers published saying that negative J is okay. And so here we go, yeah, and that's because this is the interfacial tension. Okay, so what can we do with these things? Well, this is sort of stuff that we do like at the university with the students. They play around. So this is like, okay, so this is cells which are mitosing. They get nutrients coming in. If the nutrients are not enough, they start apoptosing and making a empty sphere, right? So you start seeing like what would be an organoid, right? So some cells, they're dividing, they're growing, they're not getting their nutrients, and they divide. But it's really nice because now it's very easy to explore what happens if you change how quickly you grow, how quickly you divide, or how much nutrients you need, right? If you change the adhesions between the cells, you can get better like endo and ectoderm cells appearing, right? Here we can get, here the idea was to try to get internal structure and so forth. So these are the type of things that are very easy. I mean, this is just simple out of the box because cellular POTS model now that you have this sigma for every cell, right? You can say, oh, if you have so much nutrients, you divide into the middle, you'll have a daughter cell which will have like a new updated target area. You can have functions how these target areas grow, so you can have growth in your system, and it's very sort of intuitive how to start adding the biology. The other thing we do with it is we use it, and this is a little bit related to the talk from Pierre, we can use cellular POTS model to segment images. So we seed many cellular POTS cell babies into an image which has some type of fluorescence of the membrane, so that would be an energy cost. And they grow in a Monte Carlo simulation in the 3D image. And then you can get your cells back. So not that you want to find the total minimum here, but you have the constraints, like these energetic constraints that you're operating with, right? So this is a organoid, segmented. And that helps you now because we can start segmenting images very well. Then we already have the template to start a simulation. One idea we had also is maybe, we're still not there, if we have a dynamical image of cells moving, we could also adjust our parameters to get those type of fluctuations. How am I doing with time? Is it 20 minutes? Okay. But yeah, it's still things to do. But we have a link. We have sort of like a like a physical part here that we can think about. So what about the mechanics of tissues? So mechanics of tissues is a bit like when you're observing a tissue. So this is work by François Granet and his group. What's really powerful is looking now at how cells, at their shape, at their connections between their neighbors, and how these things might change if there is some force applied to it, right? So he's developed a lot of mathematical tools, theory, I would say, and tools, which starts analyzing mesoscopic pieces of this tissue in terms of texture tensor. And this will tell you how elongated you are, how many neighbors you are. And if you look at this dynamically, you'll be able also to do some, I would say, like bookkeeping, to be able to know what events were happening, what events did you potentially miss. So there's really beautiful work here. I would definitely guide you to that. So the origins of this is also because this is what they have been using for foam. So this is a 2D foam, right? So you make the bubbles, you let them hover on top of a surface, and you shear it. And if you have an obstruction here, what's very nice is then you can see, the movement is doing around it. So this was and you can look at this dynamically. So you can look at the shape change of your bubbles, the deformation rate. You can also look at what's happening with your topological changes, right? So for example, did you have neighborhood swapping? In the case of biology, do you have apoptosis? Did a cell die? Did it divide, and so forth? So you can have all these topological changes. This one here is our favorite for, I'll talk about it later. This is the T1, which is what you need for cell sorting, for example. You can also look at these kinematic measurements. So basically, this is it. So these two cells are in contact. They very briefly come into cross-sectional configuration here, which cells hate, and then they bang out like this. Okay, so we have this neighborhood swapping T1, and here, because he has this and this, he can actually see in what direction did it happen, so he can have a tensor for all of those entities. Okay. So now he can have a map of all these quantities. So these are these little coffee beans that you start seeing here. And this here gives us a better idea of the flows and the physics that are happening, for example, in this flow around this circle. Now, why was this so important? Because from there, they can start looking at constitutive equations for the physics of these materials. The elastic, they can look at the elastic behavior, the plastic behavior. Now, this is still foams. But one of the questions that François has in that group in France, and I think there are people in Brazil working with this also, Karina, is wouldn't it be interesting to now take our models with our cellular level parameters, put this in a tissue and start challenging this tissue with certain, maybe an experiment like this, maybe it has a flow and it has an obstruction, and to look what's happening qualitatively there on the tissue to try to see what's that emergent physics on that other scale, right? So this is something which is out there. So I just want to pose, so if people are interested, I think it's a really nice way of then thinking of, and why is that important? Because you don't always want to model everything on that level of the cell. Sometimes you just want to have like maybe an amorphous description of your tissue. It would be good enough. But you don't even know exactly how those properties would scale up, right? So it's really important to think about how we can transcend those levels. One thing we can start doing with the cellular pots model, which I think is quite fun, is we can also do measurements on it. So we could press the tissue and see what happens with the deformation of the shape. But the problem with the cellular pots model is that if you have cells here, like, we have some cells nicely connected, yeah? And I start pressing it, they're going to move. They're going to quickly undergo T1s, yeah? So they will have a shape change, but the shape change will very quickly disappear and they will pop up. And that's actually a little bit irritating because sometimes we do want to have simulations of epithelia where we know that we have very tight bonds. Or, for example, like where we have cell wall. So one thing we can do with the CPM is we can allow Monte Carlo to go naturally, but the moment that the spin flip would occur to make a T1, we prohibit. So we don't allow neighborhood rearrangement. And with that, all the deformation we apply on a tissue will be seen in the strain of the cell shape. So this is that. So this is, in this case, we are These are the texture tensors of Francois that we just implement in the CPM. And here we started pulling on the tissue, but we don't see much, right? So this is at equilibrium, and this is afterwards, so we just see a little bit of a change. And that's because we have all the T1s. I like to call it more plant-like because I work with plants, so it's not really that plant-like. Okay, so these cells here are not undergoing T1, so I'm really stretching them like enormously. But the funny thing is you can see how that response between your applied force and your response of your shape is. Okay, so you're doing the piezoelectric test on the tissue, right? So we know what our applied force is, and now we can look what's happening to the delta L over L, so the Young modulus to the Poisson's modulus for the moving cell, right? Or here, for the plant-like thing. So we see here these type of slopes. I think this slope might be important. Now, changing those parameters, we'll have different mechanical features, yeah? Exactly how that mapping goes, we don't know. We're sort of empirical at it yet. Okay, so I'm actually ending now. So my summary is everything is possible. And that I got from Rita, so it's not my... So I showed you that energy-based model, parts model is an energy-based model which describes surface mechanics. So inherently in it, because of the Monte Carlo, it has membrane fluctuations. You know, we have internal pressure, cortical tension, so those are things that we give to the model. As a result, cell shape emerges, so we can talk about what angles they will have according to those J's, and so that's all something we observe. We have cell sorting as a property. Tissue surface mechanics also comes out of it. And we can have these movements and cell shape changes due to stresses within the tissues. Now, possible extensions for modeling, which we work a lot with. If you guys want to know about this, we can talk about it. It's chemotaxis. And chemotaxis is... it's very easy, right? Because you can have a PDE running on the same grid. I mean, that's what you do, as your cells. So if here you have a membrane, here you're going to have like different concentrations. So at the moment that you do a move, that local concentration could give an increased likelihood of that movement. So basically, you're going to get a force, because you take the derivative of the concentration, and you'll move more likely forward. Or you can retract also. You'll have a backward moving. So you can easily have cells moving chemotactically in fields. Cell division, very easy to implement because you have nice shape. So the way that we do cell division is, because you have all these pixels, we calculate the first moments, right? So first, second moments, so you get like the ellipse shape of any cell, and then we usually do just perpendicular. But you can do it any way you want, right? So it's really up to you. We also have, we can transform these CPM cells into what you guys call them boids. So basically we say, oh, every cell also has like a vector, persistency vector, and it has a preference to move in that vector. So what you can say is that if you consider to do a little extension, right, there's going to be a vector here, and you just take the dot product between that vector and the persistence vector, right? So this would be like your persistency, and this is maybe like a desired move A, right? And then you see if it's closer to here, you extend more readily. If it's here, it's neutral, and if you're going to extend backwards, it's a negative term. You can do something like that. And what's very nice is you can have then update rules that, well, but if you end up like hitting something, so you might be moving like this instead, your persistency vector can be updated. So you can very nicely have self-propagating cells. It's time efficient, it's very easy to implement in cellular parts model. And obviously cell differentiation. So if I'm a cell and suddenly I see a lot of auxin, I might turn on some other genes and, well, it's not a good idea with plant cells. Let's say I'm a cell and I feel a lot of, I don't know, retinoic acid and FGF12, I'm going to express a lot of coherence, right? So you can change the physical properties according to something that you are experiencing on your field. There are also some Evo-Devo work, which are very nice with cellular parts model, where every cell has a Boolean network, and the Boolean states determine the properties, the biophysical properties. So you start getting evolution of funny traits. When cells migrate out, or if it doesn't work, if they fall off the biophysical fitness, they will just be out-selected for. So you actually get like really interesting, like you get, for example, protrusions happening, you get convergent extension, you get the formation of meristems. So there's a lot of cool things that you can use because cellular parts model is so flexible in the way that parameters map onto its behavior. Yeah, and then furthermore, because it has this mesoscale, so it's, it's a big thing, it's very easy to add different things. And I think the group in Porto Alegre has been doing that. They've actually been modeling the cell with different, like, with an explicit membrane, right? With different parts of the cytoplasm. So you can then start thinking about different subcells, right? I've done some work where I look at vacuoles in cells, which are quite cool, and cell walls, so that's one thing. And the other thing which is possible, not impossible, although we prefer to use a cellular parts model with partial differential equations underlying, because then you have these separate scales, but you can also have on the membrane moving agents. You could also have them carrying moving agents if you want. It gets a little bit messy when you lose pixels, but there is a way of doing that. So yeah, so this is the idea. It's one way of getting patterns. I hope you do think that engulfment and these things would be patterns as well. And the only thing you have to put your intuition in is understanding that you have a sticky bag which doesn't like to be pressed too hard, and that's it, right? And then all of these things sort of unleash. So that is all I wanted to tell you. For the group who's working on the dictee, we can make a container with the CPM for you guys to work on if it's interesting. Okay. Yep. Questions? Or should I?
Sorry.
No.
I have another question. Thanks, Veronica, for this great talk, great lecture, actually. I have a doubt about the POT model. I'm not sure, but you can help me. In your opinion, do you think this model can distinguish properly with its adhesion and cortical tension? Your opinion? Because J, that parameter J, is associated to adhesion between cells. And this parameter, it's, I think, linear to the perimeter of the cell, or something like that.
Yeah, so we have, this one.
Yeah, so how we can distinguish properly which is adhesion and cortical tension too, because cortical tension, the term, is linear to the perimeter too. And this parameter is linear to perimeter. So we are effectively has two terms, which is one term too.
No, the other one would be, once you differentiate it in relation to perimeter, right? But this one is squared, yeah?
But we have a linear part of this square.
Yeah. But here, this is your perimeter, and this is your perimeter. And then once you take the interfacial tension, right, when you do this here, then you just get your J here by itself, and then you get this one multiplied by P. Yeah? And you get some...
Yeah, but J is proportional to P too.
Okay. So I will tell you what my next goal is in the cellular parts model. Okay. So there is something funny about J. Okay. There is something funny. Most cases, we don't even notice it. What are we saying here? The lower the value J you put, right, the more that those cells will have a preference to be together, right? Okay. Yeah? So they're going to come together, and you're going to get this aggregation. Okay. This is what we're saying. And of course, you could invert it with a negative J. But the problem is, it's not even that. The problem is the following. If I have a sea of cells, so I have a tissue with many, many cells, and in the middle of them, I have a yellow cell. So I have lots of blue cells which like to adhere with one another. And I have the yellow cell which adheres weakly with the blue. That means it has a big J, okay? Yeah? So it's like, right? So It's, yeah, and I can measure its diffusion. It's going to diffuse around. Okay. Now I start looking at that yellow cell, its diffusion movement. It has a low J when it's in a sea of yellow cells. Okay. How do you think it's going to diffuse? It's much more adherent, but it's going to diffuse much more. Yeah. Because its energy is lower, and it's in the sea of cells, so it's actually quite happy. You know, it doesn't have that energy thing. And this is weird, because if you have a loosely adhering cell, right, you'd think it's going to move more, and if you have a very adherent cell, you think it's going to be more fixed. Yeah, so that is something which we have a problem with J. Because J is just telling you, like, actually, it's just telling you the N configuration, right? The N configuration state, which is, oh, you know, yellow wants to be with yellow. But it doesn't mean that we can immediately say that the time dynamics that it does in trajectory is correct. And why is that biologically not okay? Because if I have a really strong coherent bond with someone, it's probably more unlikely that I'm going to undo it. Yeah? There's a cost to undoing very strong bonds, which is probably bigger than, you know, if I'm weakly bond or not really having such a good chemical connection. So there is something we're thinking about, but that would mean some extension to the rule. I think the extension to the rule goes into the way we update the delta H's, right? So that we actually, you know, one thing is to say this is the minimum configuration, but the other thing is to say there's a cost to undergoing certain paths. Yeah, and that's the problem I have with J. Yeah, but to say, oh, you know, J positive or negative, this is, because there's a paper actually saying that a negative J would solve that, but it doesn't, right? Because actually by itself, it's not changing the dynamics.
My question is basically to understand how differentiate J and the cortical term, because how we can understand which drivens the cell sorting process, for example.
Yeah, so it's because it's in the interfacial tension in the end, which is the thing. Yeah. It's coming together, yeah. But how do you, okay, so this, yeah. So I agree.
Because
once you have a cortical tension, you're basically messing up your one-to-one interpretation of your J. Yeah. But this is also the case. We do know biologically that adhesion and the cortex are very, like, they talk to one, like, experimentally people were very confused because they would see these things. For example, there's this biophysicist who she has cells, and then she's able to do something where she allows there to be like a naked membrane part, because she takes away the cortex, and it blebs. You know, the blebbing, what's her name? Julie, anyway, she was in the Marie Curie Institute. She did this whole blebbing thing. I think, yeah. So this effect. So that is actually saying that you actually should look at this interfacial, in those cases, those cells, the interfacial tension and the cortex were inherently together. Like they were giving you that effective one. Once you remove this guy, right, this here is, boop, you know, had such a low cost that it blebs out. It doesn't explode immediately, but it has that effect. And then people were like, yeah, but is this adhesion, is this not? But then if you would make adhesion higher, you'd also get a different surface tension. So they didn't know. So is it cortical tension? Is it this? So I think it is mixed up. So what you have to realize biologically, you might have different ways of controlling things. But like from a surface mechanics point of view, it's going to get smeared out. You're not going to distinguish, oh, this part of the force was because of that or was because of that, right? It's just lumped together there, effectively. Okay. I don't know, does that sort of...
No.
I know, it feels esoteric,
yeah.
doesn't it?
Yeah. Sorry.
No.
Because I work with you, so I know the questions about this self-sorting process, if it's Steinberg is right, or Harris, or John is, or another one, because there's many conflicts in many debates about it. Yeah. So we can talk later. Yeah.
Any other questions or?
So in the model where each site corresponds to one cell,
Yeah.
Okay.
Yeah.
I mean, why don't you have separation? Why don't you have segregation? Because if you consider, for instance, the binary lattice gas, right? You do have separation between the species.
Yeah. So I think the problem is they want to have the separation only based on adhesion. So saying that you move blindly and only when you touch in a very close vicinity, then you stay together, right? You clump. And after you start clumping, the problem, because you're right, the problem of that is that it's very difficult to start moving without evoking other rules, a clump, like a cluster together. Because you're only doing local rules from a cell-based perspective. So it's not a gas, right? So they are envisioning these as cells. And that's where they weren't able to, like, yes, you can make things separate. You can make things separate in cell automata, yes. But if you start considering them as like, oh, we are diffusing agents which only interact through membrane contacts, because that was sort of, it was becoming very difficult to impossible, right? And that's why they thought it would be unlikely or not possible.
Okay, so there are rules which are different from the binary lattice gas, which I'm not considering there.
Exactly. And, you know, I think there was one version recently that made it work, but it's really, really complicated. And, you know, I'm not saying as a biologist, it doesn't mean like the nicest solution is the right one. But for modeling, probably, like if you're forcing your stuff to work, it's probably not really, And this is more parsimonious and still has a better sort of, you're not evoking random rules, right? So, yeah. But it's a good question.
Really cool.
It's a good question,
Any more questions? Okay.
So you're stuck with me after lunch also, right? Because I'm giving a talk.
Yes.
Good.
So we reconvene at 2 for the colloquium here.